It's biology with Mr. B. Biology with Mr. B. That's me! Hi Year 12. So I made this video to supplement the self-study PowerPoint and the learning and the tasks that you're doing in the plant transport topic. Check your emails for all the instructions to do with that. I need to review why, what and where. So why do plants need a transport system? What transport systems do they have, and where are they located actually within the organs of the plant? I need to look at how water moves through a plant. Keyword explain there. That is hard. Explaining how water moves through a plant is usually what stumps people in this topic. I need to describe factors that affect transpiration and some of the adaptations that certain plants like hydrophytes, those that love water, and xerophytes, those that live in a dry environment, what sort of adaptations they have to cope with how their environment affects transpiration. And we also need to describe translocation, which is the phloem, which is how sort of sugars and assimilates can move up and down the plant depending on need. So what I'd like to show you first is just where to find the PowerPoints and just the things for this topic. So if I very quickly go to my Tinternet, so if I click on the resources tab, which is obviously file store, you'll get to this page. You go on students, lesson resources, science, biology, whatever that is. I think we're right at the bottom, aren't we? A-level, we're in unit, module, something number 3, and transport in plants. So even if you're sat there and you don't have a transport in plants booklet because you weren't in the last week when we sort of gave them, well, when I hope Miss Hall gave them out to everybody, she says she has, it's this booklet here, Trans in Plants Theory 2019. That's the one you need to get all the tasks to go with the independent PowerPoint. The independent PowerPoint which you guys need to be working through is this first one, the one that says SAB. The one that I'm actually going to show you today is this one. It's kind of like a slimline version, very factual, very notes-based. You might also like that from a notes point of view as well. These two are a couple of old PowerPoints. You never know, they might be nice. I've actually not looked through them this year, so I can't remember. But these two are the ones we're working from. That's got your tasks and instructions, and this is what I'm doing today. Okie dokie, boys and girls. So, another little look at my face because, you know, some of you love it. So, why do plants need a transport system? So this first section, this first bit to try and clarify is why, what, and where. Why, what, and where is equivalent to sort of task 1 to 4 in the booklet. Why, what, and where. Why? Plants are generally large. I appreciate there are some little baby dwarf plants, but most plants are generally quite large. And that means they have a small surface area volume ratio. And organisms with a small surface area volume ratio find it very difficult to obtain the resources they need by simple diffusion alone. They're too big. They've got too much volume and not enough surface area, so not enough oxygen, as an example, oxygen can diffuse across their surface area to supply all their volume. So they need a specialised exchange system and they need a transport system to cope with their incredibly large surface area volume ratios. This bit here. Unlike mammals, well, not that bit there. This bit here, don't know why that's been first. Water, the only place a plant can get water is the ground, is the roots. And minerals, pretty much the only place a plant can get them is those that are dissolved already in that water, so through the roots. Because it's getting everything through its roots, and therefore it's getting everything water-wise, mineral-wise it needs from, well, the bottom part of it, it needs a mechanism to transport that water all the way to the top. So when you're dealing with the average size plant, which may only be a metre or so tall, again, that's still a metre to move. But think if you're dealing with like proper large trees, some trees can be like 100 metres tall. That means we're going to have some sort of significant transport system. Same for sugars. The only place a plant will make sugar is where it photosynthesizes, which tends to be just the leaves. So how do those sugars get to the roots so the roots can do active transport? There must be a transport system. So compared to mammals now, this oxygen idea, plants Yeah, okay, really small surface area volume ratio. They can only access water from one part of them. You'd think that, if a plant had a pump, that'd be really efficient. But plants don't need pumps. They don't need them. As difficult as it is for a plant to get oxygen inside its system, ultimately they've got such a low metabolic rate, diffusion is still fine. So what you find is, yes, they have a specialized exchange system. Think root hair cells, big surface area, lots of active transport, things like that. Yes, they've got really good transport systems. They've got tubes where water can flow, that's the xylem, and where sort of sugars can flow, that's the phloem. But they don't need a pump in those tubes. They can rely on generic movement in those tubes. Okie doke. Where? Oh no, it's not where. What? What is next? What is xylem? Xylem is a tube that transports water. And therefore, if you want to transport water all the way from the roots of the plant to the leaf of the plant, where remember, it's the leaves that will do photosynthesis and water is needed to do photosynthesis, you're going to need a tube that's waterproof. If your tube isn't waterproof, all the water would leak out of it and just go into the surrounding cells, so it would never actually get to the leaves. Now we are going to do, whenever we get back to school, the first thing we'll do is do all the practicals that you're missing out of. And I absolutely adore looking at xylem structure and function. Because one of the things you can see beautifully when you stain it is lignin. Lignin, if I just get my little red marker, lignin is an impermeable layer to water. So baby plants have no lignin, but as they get bigger and bigger, more and more lignin deposits around their xylem vessels. These xylem vessels, by the way, these xylem cells, when you're a baby plant, are alive. But the lignin, because it makes the cells waterproof, that means, yeah, right, water can't get out, but also water can't get in. It means they die quite quickly, and all the cell contents decay. But what that does, it leaves you with a long, continuous tube of dead xylem cells with no content. It leaves you with a perfect tube. We often see lignins as spirals, so they generally are spiraling sort of round and round and round and round and round the xylem tube. But they can be annular, so they could look like little rings, and they can be reticulated with a lot of broken rings. Generally, when we do the practical, we tend to find most lignin look reticulated. I'm going to presume not because that's just the plants we're using. I'm going to presume it's just because students, when using sort of knives to cut open stems, like to break the lignin. Not like to, it just happens. But because it's just not like 100% deposited with lignin, it does allow the xylem to grow and stretch as the plant grows and stretches as well. Think of it like the rings, think of it being like a spring. So if it grows, okay, they'll just stretch further apart. In some places, there is no lignin. Some places lignification is not complete, and it leaves what we call bordered pits. Now they're actually really important. Bordered pits across two adjacent xylem vessels allow water to leave one vessel and pass into the next. You might remember from doing the communicable disease topic with Mrs. Hall that sometimes plants can have a disease. So what they'll do is they might block the xylem vessels. If they block a xylem vessel, well, how does the water get to the leaf? Well, because there are bordered pits, that water can move into adjacent xylem vessels. So if there's a xylem vessel that isn't blocked, it will still allow water all the way up. Phloem. Phloem is a weird tissue. It's made of two completely different cells. Two completely different cells, but because they have the same function, they're still part of the same tissue. The phloem's job is to transport assimilates. That is a posh name for mainly glucose, although generally it's the form sucrose that sugars transport around the plant. But also think of like if there's like amino acids or certain solid base minerals, they will also be transported around the phloem. Mainly sucrose though. The two types of cells are sieve tube elements. I promise a sieve tube element is a cell, and a companion cell. Best friends. The sieve tubes, which are these, okay, they actually look like, sorry, not there, here. The sieve tubes, so I'll just draw around an entire sieve tube. They, like Xylem, they are sort of these rectangular continuous columns. The difference is they do not have lignin around the outside, so they're not waterproof, so they don't fully die. But they have no nucleus and very little cytoplasm, and that ensures you can move things along. This part here, which is where you'd expect a brand new cell to start, and maybe a brand new cell wall, it's actually perforated. It looks like a sieve, and it's called a sieve plate. So it's got little tiny holes like a cheese. Like a cheese? Now, see, my daughter's got toy cheeses. They've all got like holes in. It's like Emmendale. I was going to say Emmerdale. That's awful. I've never actually had a cheese that's got holes in. Maybe that's something, you know, if your parent goes shopping and there's no cheese left except some fancy one with holes in, maybe you can have some and tell me how it goes. Anyway, these holes allow movement of all the sap across from one sieve tube element to the next. That's the worst arrow I've ever drawn in my life, but there you go. So not completely hollowed like Xylem, not completely dead like Xylem, but very little cell parts, no nucleus certainly, very little cytoplasm. And at the end of each phloem cell, at the end of each sieve tube element, instead of a full cell wall, it's a perforated wall. So it's called a sieve plate, and it does allow the flow of sap, which would be like just fluid with those assimilates dissolved within, across. The companion cells, these guys, their job 100% is having lots of mitochondria. Like generally, that's all you need them for, to have loads of mitochondria to produce loads of ATP. And we'll come to that when we talk about translocation later. Where? Distribution of vascular tissues, always a great opportunity to ask, what is a tissue? Feel free to pause me and have a go. Tissue is, of course, it's a group of specialized cells with similar structure and function. Now, when we look at vascular tissues, we only ever look at a type of plant, not a type, these are types of plants, it's a big variety, called dicotyledonous. That is not how you actually pronounce it, that's just how I pronounce it so I can spell it. Dicotyledonous. You might see them written as dicots. They are plants that have 2 seed leaves, so they always have 2 leaves. We always look at these guys in biology rather than like monocots or tricots, only because their vascular tissue is arranged beautifully. Like it looks amazing, and it makes it very easy to label a photomicrograph. So these, just the pictures from your textbook, this is a root of a dicotyledonous plant, and this is a stem of a dicotyledonous plant. There's also ones for the leaf as well. So if I focus on the root first, we actually have this beautiful X shape in the middle. So it can be anywhere between 4 or 6 points. I think it might be 4 or 7. Textbook does say. But generally, most commonly, it's actually drawn as 4 points, which makes it look like a beautiful X. X marks the spot because the X is xylem. So all of this big bit in the middle is the xylem vessel. These bits around it, in between the arms, they're the phloem. So both xylem and phloem are located in the roots, next to each other as a bundle. Xylem is the X in the middle, or the star shape in the middle, and the phloem is in between the arms. The other main tissue to look out for is the outside part of the vascular bundle, and that is the endodermis. Endodermis. You will also find, if you think it might be in the textbook, can't quite remember, but just inside the layer of the endodermis, you will find the pericycle. That is P-E-R-I cycle. Oh, I'm proud of myself there. So this pericycle is found just inside the endodermal layer, sort of there. The pericycle is the meristematic tissue that gives rise to the xylem and phloem in that vascular bundle, and all the other tissues inside that vascular bundle. Pericycle, meristematic tissue for the root. Conversely, in the stem, we still have this lovely vascular bundle arrangement, but now there are lots and lots of vascular bundles, obviously all the way around a circle, around the medulla, the middle of the stem. It is always arranged in the same way. The xylem is on the inside, the phloem is on the outside, and in between is the meristematic tissue of the stem that gives rise, that differentiates into xylem and phloem. That middle bit is called cambium. C-A-M-B-I-U-M. Cambium. Cambium. Cool. Now, not really seen particularly well on this example. Actually, no, it is seen. Look, I've actually labelled it wrong. So what we actually usually do is whenever there's sort of three drawn, usually it wants xylem, cambium, phloem. Most commonly, you'll actually see four being drawn, and this outside layer is the sclerenchyma. I didn't realise that what they've done here is they've labelled the sclerenchyma, phloem and xylem. They've missed out the cambium, which should be in between the xylem and phloem. So sorry for that, but I hope it will make sense when I show you this next bit. So there's vascular tissues. We do call them vascular bundles. They are always bundled together. Xylem, phloem, the meristematic tissue always there as well. But then you've got other tissues like collenchyma and sclerenchyma, and they give the bundle strength to help support the plant, especially a young plant, which needs that extra bit of strength and support. Got a game. The tissue game. So if you want to play this game properly, you need me to put the question on. You then need to pause it till you get answers. Then you press play and you see if you got it right. And I just thought this was beautiful for... this is always on my little workshop PowerPoint, but I just thought it was beautiful for the current situation of you guys staying at home, probably all by yourself, unless you happen to be a twin, in which case that's hilarious that you have to stay together during this time. Either way, if you are feeling lonely, don't be. Demodox, the little cute little fate, is a type of mite that lives on your face. There's hundreds of them on your face right now. Best friends. So, first photomicrograph. Again, if you want to play this properly, you've got to pause it when you see the picture and then guess. So, this is a picture of a root. Can you see the X in the middle? That X, which is represented by B, is the xylem. In between the arms, here, and here, are the phloem. And this outside circle layer is the endodermis. Question 2. It's a leaf. Okay, so I've added some extra tissues here. The ones that kind of matter from the xylem phloem point of view. C is the xylem. The xylem is always found on top of the leaf, on top of the vascular bundle in the leaf. And D is the phloem. There will be a small layer of meristematic tissue obviously in between these, but we don't have a certain name for it. A is your palisade mesophyll cells. You recognise all the rectangular bundles, big surface areas. B is the spongy mesophyll cells with all the air spaces, so gas exchange can take place. E, which is this very, very top layer, that is your upper epidermis. Upper epidermis, the thing that's usually lined with a waxy cuticle. So here's the next little question. How have they made that image? Because it's not just one colour, is it? They've clearly stained it. And it is a light microscope graph. It is a light microscope producing that photomicrograph. Yet we've got different colours for different cells. There's a different colour for the xylem and the phloem. There's a different colour for the palisade mesophyll to the sponge mesophyll. How have they made it so it's got different colours? Your answer, boys and girls, differential staining. If you can't remember what that was, go back to cell ultrastructure topic when we talked about stains. differential staining. Same stain, but stain things different colours. I can tell you now, the stain they've used for that is toluidine blue. Toluidine blue is the one we will use in a practical when you get back. And what advice would you give to a year 11 trying to stain a sample for the first time next year? Do you remember the advice I gave you at the start of year 12? Don't just splash it on with a pipette. Put your few drops on this side of the glass slide and then spread it across the sample. Maybe using blotting paper. That way ensures everything gets covered. And you haven't got just loads of stain in the middle but not towards the side of it. And also, would you give advice about avoiding air bubbles by lowering the cover slip gently, starting at sort of 45-degree angle and lowering it gently with a one of those little pointing mountain needles, those little pointer poker things. Last question. This is the stem. So this one is actually it's far easier to see the fact we've got 4 layers in the vascular bundle. In a photomicrograph, you probably will see 4. The very inside layer has to be the xylem. Xylem's always on the inside, facing the medulla of the stem. The next layer along will be the cambium, which is the meristematic tissue that gives rise and differentiates into the xylem, and the C, which is the phloem. And on the outside, you have this strengthening tissue called sclerenchyma or sclerenchyma. Okie doke. What I've just gone through there, that is the first chunk of that topic, the why, what, and where. And that science will take you through, hopefully, tasks 1 through to 4. The next bit is the bit that people find very tricky. How water moves through plants and transpiration in general. So again, the reason why I'm making this is so you've got it permanently. You can keep rewinding, replaying it whenever it makes sense to. First job, water movement through plants. When water moves through plant cells, it can move 3 different ways. It can either move in between all the cell walls. So notice how it never actually goes through any cell membranes here, which means it doesn't move by osmosis. Osmosis needs a partially permeable membrane. That pathway there, A, is called the apoplast pathway. So it moves between the cells, between the cell walls. Pathway B, where we're actually moving into the cell, so this is definitely osmosis, and into the cell and just across cells via the cytoplasm. That's the symplast pathway. And C, where it's going through all the vacuoles, that's the vacuolar pathway. A water molecule can move any of those pathways. It can move a mixture of those pathways. It could go like the symplast pathway for this first cell, then it could go into the vacuolar pathway, and then maybe it could go out into the apoplast pathway. The water doesn't get to choose which way, but there are a couple of mechanisms plants have to ensure water goes certain directions and certain pathways. And one of them we look at next. Okie doke. Definition in red. The transpiration stream is the movement of water from the soil through the plant to the air surrounding the leaves. So through the plant, what does that mean? It means through the root, through the stem, through the leaf, and out to the air. Water enters roots by osmosis at these root hair cells here. And of course, dissolved minerals will be moved as well. This water entering the root hair cell will move this way along the cortex. All this bit here that I'm drawing in red is the cortex. The cortex generally of most organs usually means the outside part of them. Medulla usually means middle. And it moves all the way towards the xylem. The xylem are these 2 tubes here on the far right of this diagram. And it moves down a water potential gradient. It's moving by osmosis. Except obviously the water going the apoplast pathway, because if you're not going across a cell membrane, it is not osmosis. So water can move by any of those pathways, symplast, vacuolar, apoplast. But here's where it gets interesting. Think of this. This is where it starts to get weird, a bit more tricky, so you've got to really think. You want 100% of the water that entered the root to end up in the xylem. You don't want it moving this way. You certainly don't want it moving backwards. You have to keep it moving into the xylem, because that's the only way it's going to get up the plant and to the cells that need it. The only way we can do that is if we maintain and also create an incredibly steep water potential gradient. So we've always got a really high water potential gradient here compared to a much lower one here. And every single adaptation I'm going to talk about now, that adaptation is to meet and create that gradient. First one then. This yellow thing here, which is along the endodermis, so along that sort of outside circular part of the vascular bundle, it completely and utterly blocks the apoplast pathway. So if you've been travelling like this in between all the cell walls, not actually going through the cells, you'll get to the Casparian strip and you'll just stop. And the only way that water can now move is into the cells, into the symplast and vacuolar pathways. Think what you've done there. You've purposely moved more water into these cells here. So these cells have much more water than they used to, because all the water from the apoplast pathway, this bit, has moved in. So I've now got a high water potential here. Therefore, I should get more osmosis this way into the xylem. I'm creating a steeper concentration gradient, water potential gradient. The plasma membrane as well. Ooh. The plasma membrane of the endodermis here also has transport proteins which do active transport and pump mineral ions from the cytoplasm of these cortex cells into the medulla cells and the xylem. So again, think what you're doing. By pumping mineral ions this way, you're lowering the water potential here and therefore creating a steeper concentration gradient. The Casparian strip as well, once water does get this way, it can't just escape back down the apoplast pathway because you've got the Casparian strip blocking it. The water Casparian strip is impermeable, so water can't get across. But it can get through the cells. It just can't get round the cells. So that's how water can actually move. That's how it actually gets into the root. And once it gets into the root, it needs to move upwards. Because the xylem is hollow, this can't be osmosis. There is no osmosis in the xylem. Instead, it moves by mass flow. Mass flow is generally, we usually use the word mass flow to indicate movement where there is no diffusion, osmosis, active transport. So a really nice example of mass flow is blood moving around your body. It's moving, it's not diffusing around your body, it's not osmosing or active transporting, it was pushed round, so it's moving around by a pressure gradient, mass flow. There are three things that help water move up the stem by mass flow. One, root pressure. Because you're constantly moving water into that root medulla, so across the endodermis, into the medulla of the root, into the xylem, the pressure in that very bottom part of the xylem and the root builds up and up. So obviously some of that water moves into the xylem and it can move up a few centimetres or so. So not enough to get it all the way to the top of the plant, but it's enough to get the movement started. Here's the really big one. Transpiration pull. You might remember that water, one of its properties, is very cohesive. Water molecules are very good at sticking to each other. And they can do that because of their weak positive and weakly negative elements to the hydrogen-oxygen part, which allows them to form hydrogen bonds. That means the water molecules inside your xylem are held in a chain. And it doesn't matter whether your xylem vessel is 20 centimetres tall or 100 metres tall. The water will be held in a chain. What means is, when root pressure happens and a little bit of water gets forced upwards into the start of the root, because they're all connected by chains, that actually pushes all the water molecules in the entire in the entire xylem upwards as well. When water moves by osmosis in the leaf, maybe you get transpiration, you actually get water vapour leaving the leaf, and water moves into the leaf to replace it. Well, because they're all attached to each other, if one water molecule moves to the leaf, they all move upwards. Transpiration pull, cohesion tension theory. And this is also why the xylem desperately needs lignin. When it's under such tension, the lignin is there to prevent the xylem vessels from collapsing. Final point. Adhesion, capillary action. Water molecules are sticky to each other, but also to other things. The water can be attracted and form hydrogen bonds with the walls of the xylem. That's called adhesion. Cohesion is where it's two of the same molecules. Adhesion is where it's two different molecules. And because they're attracted to those walls, it ensures it doesn't just randomly fall back to Earth. It ensures the water can keep moving upwards and only upwards. And because xylem vessels are so narrow, even those adhesion forces can pull water up the sides, can pull it up and up and up. Now, if you're looking at the big answer for this, of why does water move, this is your big answer. This causes huge amounts of mass flow. If there was no transpiration pull, there would be no plants bigger than a metre. Because of transpiration pull, you can get giant plants and the xylem vessels still work perfectly. So what actually is transpiration? We've got a second little red definition. The loss of water vapour from the aerial parts of a plant, mostly through the stomata in the leaves. The definition for transpiration, actually how it happens, is exactly the same as it was at GCSE. The water, which was in the xylem, enters the leaf by osmosis. So the water is now in here, in the spongy mesophyll bit of the pathway. That water, because this is a leaf and light's shining on it, the water might evaporate. Evaporated water, which is gaseous water, will move by diffusion down a water vapour gradient out of the stomata, out of this little pore here called stomata. These two green cells here, they're called guard cells. One of the little practicals we'll do when you get back is we'll investigate stomata, we'll look at them properly, we'll do a bit of maths with it as well. Environmental factors that affect transpiration. Again, these were all discussed at GCSE. Light intensity. The higher the light intensity, the more stomata open. The more stomata open, great, the more gas exchange, more CO2 for cells. Because imagine, if there's more light intensity, you should be able to do more photosynthesis. So you want more carbon dioxide moving in. But conversely, you'd have more water leaving by diffusion. More water would evaporate, more water would leave by diffusion. So it's kind of a win-win. Yeah, you're going to get more CO2, but also you're going to lose more water. So you've got to make sure you have enough water and you're not in a dehydrated position. Temperature. The higher the temperature, the more evaporation. The higher the temperature, the faster the diffusion. Humidity. By humidity, we effectively mean how dry the air is. If the air is very humid, that means it's very wet. If the air is wet, you're not going to have much of a concentration gradient. You're not going to have much of a concentration gradient between inside and outside of the plant. So less transpiration will take place. If the air is less humid, that means it's drier. That means there will be a steeper concentration gradient between the plant and the outside, so you would get more transpiration. Air movement, wind. If it's very windy, all the water vapour, which is like here, if it's just diffused out the plant, all of it will be taken away by the wind. And that ensures you maintain a steep concentration gradient because all that water molecules get moved away from the stomata. If there was no wind and it just diffused down and then the water vapours just stayed outside the stomata, well, you're making the outside stomata very wet, therefore reducing the concentration gradient. And therefore, the lower the concentration gradient between inside and outside the plant, the less transpiration, less diffusion will take place. Final one is water availability. If there is no water, the stomata simply close. Good. the big practical that we do when you get back is actually measuring rates of transpiration using a potometer. We are going to be able to make our own little potometers, which are quite cool, but also we'll get Jill to make the big fancy ones. A potometer's job is to measure the rate that water is taken up by a leaf. And that gives us a reasonable estimate of transpiration. Because if water is taken into a leaf, that implies that water has been lost from a leaf. There's a couple of different ways how this can work, but most commonly, what you might do is you might put a little air bubble in the potometer tubing. And you'll leave it a certain, say you might leave it an hour, and you'll just see how far along the air bubble has moved. And you'd measure how far it's moved, and if you know the dimensions of the tube, you can work out actually the volume of water that's moved in that time. That's one way to do it. There's other ways to do it, but that's classically the way transpiration might work. Some transpiration sort of bits, for example, like the little one we're going to make will be what we call mass potometers. Mass potometers, what you do is you'd get your plant, you'd get mass of it, you'd leave it for a while, ensuring you just sort of leave it for a while and then remass it and measure the difference. And the difference would usually be the volume of water that's escaped. If you want valid results, because I appreciate in the booklet there is a task to do with like this sort of experiment. If you want valid results, make sure you set your plant up under water. It ensures there's no air bubbles inside the apparatus. It ensures it's all water, no air. Ensure that your shoot is healthy. Nice little control variables here is like, you want the same size, same species of plant, same number of leaves, same sort of health. You don't want one plant that's got a disease and another one that's healthy. Try and cut your stem underwater, and that prevents air entering the xylem. And you'd cut that stem at an angle. That angle would ensure it's got the larger surface area as possible in contact with the water, and therefore can uptake as much as it needs. You also want to ensure you have dry leaves. That ensures you're not affecting the concentration gradient between outside and inside the leaf. So dry leaves only. Okay. So that was the end of the second sort of part to this topic, and it was the hard part. The transpiration stuff, especially the moving water, is always seen as the hard bit. The third bit is to do with adaptations. What adaptations do certain plants have to cope with either a very, very aquatic based environment, like this hydrophyte here, this is a water lily, compared to those that live in a very dry environment. This is the leaf of a marram grass. So marram grass first. So again, this is in a very dry environment. We call these xerophytes. The leaf is rolled into a circle. This way, the only way air can actually get to the leaf, in and out, is that bit there. It means that all this air here becomes very, very humid because it's trapped and it can't escape. The very outside layer is a very thick, waxy cuticle that stops any water escaping out that way by accident. The stomata are only found on this inner bit here. They are only found on the inside bit. And in particular, they are usually only found in the pits. And yep, these are very much hairy pits as well. Yummy. The stomata are only being found in these pits. Again, think there is going to be no wind movement. No wind movement. So we're never going to have a steep concentration gradient of water, which means you're not going to lose that much water by transpiration, which is perfect in a dry environment. The hairs as well help reduce that air movement. The spongy mesophyll, so inside, is very, very dense with very few air spaces, and it means very little evaporation can take place as well, even inside the leaf. So marram grass, really, really clever at stopping water from escaping by transpiration. It ensures the water stays in the plant. Water lilies. So these little pockets here, by the way, rather than just being sort of like, they are sort of just empty air spaces. Really large air spaces. I mean, even in that diagram there, you're talking the air spaces are taking up probably 50% of the leaf. The stomata are only on the upper surface of the leaf. Just think for a second why the stomata wouldn't be on the lower surface of a water lily. Water lilies, if you don't know, are the ones that float on the water. So if the stomata are on the lower surface, that means the stomata is in the lake. You're not going to get much air movement in a lake. If they're on the upper epidermis, that means, yay, they're facing the air. You can actually get gas exchange between. Even the leaf stem has loads of air spaces as well, and that allows oxygen to fuse quickly to the roots for aerobic respiration. Last little bit for this. Water cannot evaporate in high humidity, and that's a hydrophyte's problem. There's so much water around it, usually the air is really humid as well. So hydrophytes sometimes struggle to transpire. You might think, well, why does that matter? They've got so much water, it doesn't matter. You know, why do you want to transpire? We've been talking about xerophytes here saying, don't transpire. Oh my God, keep all the water in. But you have to transpire. Because it's not just water moving up the xylem, it's the mineral ions diffused within as well. So diffused within, dissolved within as well. So the only way to get new mineral ions like nitrates, phosphates into a plant is transpiration and the transpiration stream. So what some of these sort of hydrophytes have done, quite a few of them have hydathodes. They are little structures which release water droplets and may evaporate them from the leaf surface. If ever you wake up in the morning and you've got a grassy field, it's got like the little droplets of dew. Okay, they've been caused by hydathodes. They always look like little stalks just sticking out the plant, which are releasing little bits of water drops at a time, which can evaporate from that leaf surface, and therefore taking water with it, encouraging the transpiration stream. Okay, the only thing I've not mentioned for this idea is actually on this first slide bit here, which I've now just crossed out, sorry. Many large air spaces. That means the leaf also floats on the surface of the water, ensuring it's always in contact with air, and therefore gas exchange can always take place. Okay, final little practical that we are going to do when we get back, but I have to mention this because there was a question on the AS paper that we gave to our current year 13s last year when they were doing sort of their summer exams, and it was just asked how would you, they were asked for sort of, I think 4 marks, how would you measure the density of stomata on a leaf? And although they had all done it in the classroom, because we hadn't necessarily mentioned or made a big deal of the word density of stomata, they didn't have a clue what to do. Like, some very clever mathematicians gave me some really lovely formulas, so like they might calculate density like in a physics sort of way, but when we say density of stomata, we simply mean how many stomata are there per millimetre square. So it's usually, you might say, if you want to just how many stomata are, you might say abundance, but because you're doing it per millimetre square, you would call it the density of stomata. And using a leaf, a microscope, and the lovely eyepiece graticles, which I know you're all big fans of, you can do it. Pop a leaf under a microscope. Count how many stomata you see in the field of view. Measure the area using the eyepiece graticule. Measure the area of your field of view. Remember, a circle, the area is pi r squared. And then measure the area of the leaf. If you saw 4 in 1 millimetre squared, and you know that your leaf is 100 millimetres squared in area, well, 4 times 100 is 400 stomata in the entire leaf. It would be an estimate, but you'd be surprised actually how accurate you can be with your estimates. So we are gonna do that experiment when you get back as well, though. Okay, so this bit is now the final part of plant transport, translocation. Active loading. So we get to see how now, where the sieve tube elements, which are the names of the cell that makes up the tube, and the companion cells, how they work together to move sucrose into the sieve tube elements and move sucrose out of the sieve tube elements. Translocation does have a definition, it's in red. The movement of assimilates throughout the plant. First little keyword. If you have a part of the plant that loads assimilates into the phloem, into the sieve tubes, you call that part a source. A source. You might presume, if you think sort of like if glucose, sucrose is going to be our main assimilant, you might presume that most sources are at the leaf, because the leaf is where all the glucose gets made. So clearly it must be putting it into the phloem at the leaf, so the leaf is a source. Usually that's true, but it depends what time of year it is. If it's winter and the leaf isn't photosynthesizing, it's not a very good source, is it? So if you see the word source, don't get hung up that it has to be the leaf. It usually is, but it doesn't have to be. During the winter, the main source in a plant is the roots, because the roots have been storing like all the starch, so they can break it down into glucose when needed and put it into the phloem sieve tubes to move it up the plant. And this is now how we differ. Sieve tube elements can move the assimilates up and down the plant. It all depends on the pressure gradient, because it all depends on mass flow. So active loading. How do we actually get assimilates into the sieve tube? First, I told you these companion cells have loads of mitochondria. They have the mitochondria to make lots and lots of ATP. And the first thing that ATP helps to do is it actively transports hydrogen ions out of the companion cells into the surrounding tissue. And therefore you create a concentration gradient of hydrogen ions. There is a high concentration of hydrogen ions outside the companion cell and a low concentration inside the companion cell. So hydrogen ions are going to want to return by diffusion. But here's the really clever part. Being a polar ion, obviously it's not going to be able to just diffuse straight away across the non-polar fatty acid tails of the phospholipid bilayer. Instead, it will need to do facilitated diffusion. The special protein that does facilitate diffusion in a companion cell is actually a special co-transport protein. It's a protein where, the hydrogen ions can diffuse across, but they can only diffuse across if they take a molecule of sucrose with them. How clever is that? They've used ATP to actively transport hydrogen ions out, and then the only way hydrogen ions can get back in, because it's a polar molecule, so it has to go across a protein because it's facilitated diffusion, the only proteins they have in their membrane are co-transport ones where the hydrogens can only go back if they take a molecule of sucrose with them. I generally love that. And now we've got sucrose in the companion cell, and it will be able to just diffuse across the companion cell into the sieve tube element. And again, that would just be down a normal concentration gradient of sucrose. Because if you're moving sucrose into here, obviously it's going to be high, and the sieve tube element must be low. Special little word, it's diffusing through what we call plasmodesmata. Now, the likelihood is you might not have come across plasmodesmata yet. You may have done when you did communicable disease. Either way, plasmodesmata are gaps, are basically little connection points between cells made of cytoplasm. So it's little strings of cytoplasm that can connect a couple of different cells together, perfect for cell signaling, communication between cells. So that's active load. That's how you get the sucrose into the sieve tube elements. So next step, how do the assimilates move from one sieve tube element to the others, and how does the assimilates get into the cells that need it? So the part of the plant that loads assimilates into the phloem tube was called the source. We've done that. And then those assimilates will move by mass flow. It will move because of a difference in hydrostatic pressure between the source and the sink. The sink is the part of the plant that removes assimilates out of the phloem sieve tube. So the sink is the opposite of source. So why does this happen? Why do we have a pressure gradient? If sucrose is entering here, so I've got lots of sucrose, what you've done is you've lowered the water potential of the sieve tube element. So for every sucrose that moves into the sieve tube element by diffusion, water follows by osmosis. So lots of sucrose and lots of water. What you've done then, because you've pumped I say pumped, it's all passive, diffusion osmosis. Because you've got lots of sucrose and water now inside that tube, it also has a very large pressure. And this pressure here at the source will always be higher than the pressure at the sink. Again, there's a reason for that. At the sink, the sucrose gets removed, again, by diffusion, to cells that need it. Cells that are respiring obviously have little to no sucrose, so it will move by diffusion. And water, because again, if you're moving sucrose this way, you're making these cells have a lower water potential, so water moves by osmosis to follow it. So this will always have a very high hydrostatic pressure, and the sink will always have a very low hydrostatic pressure in comparison, which means we always get the mass flow of assimilates. So last thing to remember, the source isn't always a leaf. Assimilates can move up or down the stem based on needs. So in the winter, it's usually the source is actually at the root and the sink will be up here at the leaves. So it can go either way. It depends where sucrose diffuses in or out. It depends where that sucrose, where those assimilates are needed. Okie doke. That is what I've got for you for plant transport. So double check your emails in terms of actually the work I want you to do. I will be putting on a webinar or two. I'll probably put one on actually over Easter if you have any questions as you go. And again, there'll definitely be one the Monday after Easter. It'll be my turn, not rather Miss Hall's. Again, to go through bits of the work, to go through the tasks, to ask as many questions as you can. That'll be the point of that webinar. So thank you very much for indulging me. I hope you've had lots of fun. Peace out.